Bismillahirrahmanirrahim. Assalamu alaikum. Now, today I welcome all my students of second year MBBS BDS regarding the fourth lecture of endocrinology. Now, what we are going to do today is about the second messenger mechanisms for mediating the intracellular hormonal functions. Now, the hormones they exert the intracellular actions to stimulate the formation of second messenger cyclic AMP inside the cell membrane. And later on, this cyclic AMP is going to lead to various subsequent intracellular effects of the hormone. The only direct effect the hormone has on the cell is to active a single type of hormone receptor. Cyclic AMP is not the only second messenger system used by the different hormones. There are two other specially important ones, which are like calciamines and associated calmodulin, the products of membrane phospholipid breakdown, etc. So, we should be knowing about the hormones which use the adenyl cyclase, cyclic AMP, second messenger system. They are ACTH, they are epithelial cells, angiotensin 2, calcitonin, catecholamines, corticotrophic releasing hormone, follicle stimulating hormone, glucagon, HCG, luteinizing hormone, parathyroid, which is the parathyroid hormone, secretin, somatostatin, TSH, vasopressin. Now, few of the many hormones that use the adenyl cyclase cyclic AMP mechanism to stimulate their target tissues binds the hormones with the receptors to allow the coupling of receptor to a G protein. Now, if the G protein stimulates the adenyl cyclase cyclic AMP system, it is going to lead to known as the G protein stimulating protein, thence catalyzing the conversion of small amount of um, ATP into CAMP inside the that is the cyclic AMP inside the cell. Now, which is going to lead to the activation of the CAMP related or cyclic AMP dependent protein kinases, and kinases they lead to phosphorylation of specific proteins in the cell, triggering the biochemical reactions which ultimately lead to the cell's response to the hormone. Once the cyclic AMP is formed inside the cell, it usually activates a cascade of enzymes. Now, first one enzyme is activated, which activates a second enzyme, which activates a third, and so forth. The importance of this mechanism is that only a few molecules of activated adenyl cyclase immediately inside the cell membrane can cause many more molecules of the next enzyme to be activated, which can cause still more molecules of the third enzyme to be activated, and so forth. Now, cyclic AMP mechanism by which many hormones they exert their control of cell functions, ADP, adenosine diphosphate, ATP, adenosine triphosphate. Now, see how the protein kinases they are being activated, leading to protein formation, which ultimately going to leads to the cell's response. This diagram is very important because this is very much self-explanatory. You can see how the hormone is binding to the G protein, and then G protein is going to lead to the activation of ATP to cyclic AMP, and later on the cyclic AMP is going to act on the protein kinases which are phosphorylated, and there is formation of proteins, uh, and they in turn leading to the cell's response. Now, if the binding of hormone to its receptors is coupled to an inhibitory G protein denoted GI protein, adenyl cyclase will be inhibited. So, reduces the formation of cyclic AMP and ultimately leading to an inhibitory action in the cell. Depending on the coupling of the hormone receptor to an inhibitory or a stimulatory G protein, a hormone can either increase or decrease the concentration of cyclic AMP and phosphorylation of key proteins inside the cell. Different functions are elicited in different target cells. such as initiating synthesis of specific intracellular chemicals, causing muscle contraction or relaxation, initiating secretion by the cells, and altering the cell permeability. A thyroid cell stimulated by cyclic AMP forms the metabolic hormones thyroxine triiodothyronine. The same cyclic AMP in an adrenocortical cell causes secretion of the adrenocortical steroid hormones. In epithelial cells of the renal tubules, the cyclic AMP increases their permeability to water. Cell membrane phospholipid second messenger system hormones that use the phospholipase C second messenger system, angiotensin 2, catecholamines, gonadotropin releasing hormone, growth hormone releasing hormone, oxytocin, thyrotropin releasing hormone, vasopressin. Some hormones they activate the transmembrane receptors that activate the enzyme phospholipase C attached to inside projections of the receptors. Now, this enzyme catalyzes the breakdown of some phospholipids in the membrane, which is phosphatidyl inositol diphosphate, into two different second messenger products, that is inositol triphosphate and diacylglycerol. The IP3 mobilizes the calcium ions from mitochondria and the endoplasmic reticulum, and the calcium cells then have their own second messenger effects, such as smooth muscle contraction and changes in the cell secretion. This is how this second messenger system is working. Diacylglycerol, the other lipid second messenger, activates the enzyme protein kinase C, which then phosphorylates a large number of proteins, leading to cell's response. In addition to these effects, the lipid portion of diacylglycerol is arachidonic acid, which is the precursor for the prostaglandins and the other local hormones that cause multiple effects in tissues throughout the body. C. How the peptide hormone is going to bind to receptor, activating the G protein, leading to the lipid membrane breaking down of phospholipase C, going to affect on the phosphatidyl inositol diphosphate, leading to IP3 DAC formation, IP3 leading to the uh, release of calcium ions, whereas diacylglycerol is leading to the kinase action, forming the protein, um, leading to cell response. Calcium calmodulin second messenger system. Another important second messenger system operates in response to the entry of calcium into the cells. Calcium entry may be initiated by changes in membrane potential that opens the calcium channels, a hormone interacting with membrane receptors which opens the calcium channels. On entering a cell, calcium ions bind with the protein calmodulin, and this protein has four calcium sites. And when three or more of these sites have bound with calcium, the calmodulin changes its shape and initiates multiple effects inside the cell, including activation or inhibition of protein kinases. Activation of calmodulin-dependent protein kinases via phosphorylation, activation or inhibition of proteins involve the cell's response to hormone. One specific function is to activate myosin light chain kinase, which acts directly on the myosin of smooth muscle to cause the smooth muscle contraction. And the normal calcium, as we all know, is around 10 raised to power 8 to 10 raised to power 7 moles per liter, which is not efficient, not enough to activate the calmodulin. And but when this calcium concentration rises to like 10 raised to power minus 6 to 10 raised to power minus 5 moles per liter, enough binding occurs to leading to intracellular actions of the calmodulin. And this is almost exactly the same amount of calcium ion chain that is required to lead to the activation of troponin C, leading to the skeletal muscle contraction. Then, last but not the least, we should be knowing about the hormones which act mainly on the genetic machinery of the cell. And what are they? They are the steroid hormones leading to increased protein synthesis. Another means by which hormones they act specifically, the steroid hormones secreted by adrenal cortex, ovaries, and testes is to cause the synthesis of proteins in the target cells. Now, these proteins then functions as enzymes, transport proteins, or structural proteins, which in turn provide the functions of the cells. Now, the sequence of events is as follows: that when the steroid hormone diffuses into cell membrane, it leads to enter the cytoplasm and it binds to a specific receptor protein in the cytoplasm. Now, then this receptor protein complex diffuses into the nucleus, and inside the nucleus, it is going to bind to specific points on the DNA strands, leading to activation of messenger RNA specific genes, which are going to diffuse into cytoplasm to promote the different steps of synthesis of proteins. That is, the new protein formation occurs. To give an example, we have the aldosterone. How? By binding the specific mineralocorticoid receptor, within 45 minutes time, it leads to the reabsorption of sodium from tubules and potassium secretion into tubules. The full action of steroid hormone is characteristically delayed for at least 45 minutes up to several hours or even days. This is in marked contrast to almost the other hormones, which are like spontaneous action. Thyroid hormones, they increase the gene transcription and in the cell nucleus. The hormones like thyroxine, iodothyronine, T4, T3, they lead to increased transcription by specific genes in the nucleus. How? This is by the receptors. They are activated transcription factors, which are located within the chromosomal complex, and they control the function of gene promoters. They activate the genetic mechanisms for the formation of many types of intracellular proteins, like 100 or more. Many of these enzymes that promote enhanced intracellular metabolic activity is virtually all cells of the body. And once bound to intranuclear receptors, thyroid hormones can continue to express this control function for days or even weeks. So this is all about the um, today's lecture, and we are done with the introductory part of endocrinology. And stay tuned, stay blessed. Allah Hafiz.